Hello everyone, welcome back to the week 8 question answer session of the experimental biochemistry course. This is coordinated by Professor Shagota Dasgupta and Professor Shomode from IIT Kharagpur, and I am Bias Mukherjee, a research scholar from Dr. Shomode's lab, and today I will be discussing with you the topics enzyme kinetics. So in today's lecture, we will be talking about enzymes, the different properties of enzyme catalysis, then enzyme kinetics. We will talk about allosteric enzymes and simple enzymes which follow Michaelis-Menten equation, and then we will see the different plots in enzyme kinetics like Michaelis-Menten plots and Lineweaver-Burke plot, and we will also solve a lot of MCQ questions related to these topics. So let us start talking about enzymes. So enzymes are biological catalysts. So they are biological molecules that catalyze biochemical reactions with very high specificity. So majority of enzymes are made up of proteins. However, there are a small group of enzymes that are made up of RNA, and these are known as ribozymes. And if you can see in this diagram, this is also a proteinaceous enzyme. So this proteinaceous enzyme is having this tiny active site to which you can see this small molecule bind. Now this small molecule is the substrate for this particular protein molecule, and it is going to bind to the active site of the enzyme. So enzymes play an essential role in almost all biochemical processes ranging from metabolism and digestion to DNA replication and cellular signaling. They accelerate chemical reactions by lowering the activation energy required for the reaction to occur. So enzyme activity can be regulated through various mechanisms including allosteric regulation, covalent modification and feedback inhibition. So we will look at all of these properties of enzymes and the mechanism of their regulation in details as we move through the presentation. So if you can see in the next figure, this particular substrate is now bound to the enzyme. And what happens? There is very high specificity between the enzyme and the substrate, and it has bound to the active site, and now this particular enzyme is going to work on the substrate and convert it into a product. So understanding enzymes allows the scientists, why do we need to study about enzymes? So we have understood that enzymes are very important biological molecules which acts as catalyst in our physiological life processes. Okay, so all the reactions that are happening in our body, enzymes are basically acting as catalyst and lowering the activation energy so that these reactions occur quickly. All right. So, but then how are enzymes important? Why is the study of enzyme so important? So, let's see. Understanding enzymes allows the scientists to unravel the mechanisms underlying cellular metabolism, disease processes, and drug interactions. Insights into enzyme function provide opportunities for the development of novel therapeutic strategies, diagnostic tools, and biotechnological applications. Ultimately, the study of enzymes not only enhances our understanding of biology but also drives advancements in medicine, agriculture, and industry. So, this tells us that the subject, the study of enzymes, this spreads far and wide into different disciplines of science, and therefore it is very interesting and important to study about enzymes, to learn about their mechanism of action, to learn about their different structural conformations, the different mode of action, their inhibitors, and all of these different studies can help us in disease biology and uh, drug interactions and so on. So there are many cases in which small molecular drugs are developed and these are made to bind to the receptor. So let's say the receptor is a protein or an enzyme. So in that case, this study of enzymes would be very, very crucial. Besides that, proteins or enzymes, they are also therapeutic targets in many cases. So then again, these proteins will be engineered, and there is enzyme engineering, there is metabolic engineering, there is protein engineering. So all of these disciplines work with engineering the interactions and the structure and function of enzymes, and so that we can attain the desired goal. All right, so let us move on. And to understand enzymes, you need to know about a few terms. Okay, so let us get familiar with these terms now. So firstly, if you look at this figure, you can see that there are two distinct colors. So this blue here, this tells you that this is an incomplete enzyme which is functionally inactive, and this is called the apoenzyme. So when does this enzyme become functionally active? When a coenzyme comes and binds to it to form a hollow enzyme. So hollow means whole, that is complete, and this hollow enzyme is a functional biologically active molecule, and this coenzyme is often a organic molecule. It is an organic molecule. It is often derived from vitamins that assist enzymes in catalyzing reactions by carrying chemical groups between enzymes and their substrates. Okay, so NAD plus, for example, nicotinamide dinucleotide, is a coenzyme involved in many oxidation reduction reactions. Cofactors, on the other hand, this is a small cofactor. This is often a metal ion. So they can be inorganic ions or non-protein organic molecules that are required for the proper functioning of an enzyme. Now, if a cofactor is bound to an enzyme using a covalent linkage, then it is called a prosthetic group. Okay, so zinc ions or Zn2 plus can serve as cofactors for certain enzymes involved in the DNA replication. Besides this, you know that Mg2 plus is required for the functioning of chlorophyll molecule. Once again, for hemoglobin, we know that iron is very, very essential, right? So then we look at the other terms associated with enzymology. They are apoenzyme and holoenzyme that we learnt about just now. So the protein portion of the enzyme that lacks is cofactor or coenzyme, and therefore it is biologically inactive. This is called the apoenzyme. So this is like a car without gasoline, that is, it cannot function, right? And then what happens? Holoenzyme is the complete or the active form of an enzyme, and this is made up of both the both the apoenzyme and the coenzyme. So an example is DNA polymerase, which consists of both uh, the protein component and the magnesium ion component. Okay, so now let us talk about the active site of an enzyme. So if you look at this figure, on the top you see that the when the enzyme is not properly folded, then what happens? The amino acids are present in it in the primary structure, but then because of insufficient folding, they are not in specific, in correct orientation. They are not close to one another. The charge is not sufficient, and therefore this is not uh, this is not going to allow the substrate or any kind of competitive inhibitor to bind to it, and therefore this is not active. So see, the amino acid side chains, they are not in a specific orientation in an unfolded protein, and therefore it lacks any activity. But after folding, if you see, all these amino acid side chains, they are having definite charges and they are aligned in specific orientations that is going to be complementary to the chemical groups on the substrate, so that the substrate can come and comfortably bind. All right, so this is a folded form of the protein which is functionally active. So see here, active site of an enzyme is a specific region or pocket on the enzyme surface where the substrate binds and undergoes a chemical reaction. It is typically a groove. So if you look here, this is like a hollow space inside, right? So this is like a groove or a cleft, okay, where the substrate binds and undergoes a chemical reaction. It is typically a groove or cleft formed by the arrangement of amino acids in the enzyme's three-dimensional structure. So, most importantly, the specific orientation, chemical groups and the charge of the residues in the active site make up the catalytic center for binding to the specific substrate during enzyme catalyzed reactions. So, in biology, this specificity is the term that we come across whenever we study enzymes and their substrates. Okay. So we will move to the characteristic features of the active site of an enzyme. So the first thing is specificity. So the orientation and the position of the chemical groups, also the charges of the chemical groups of the residues making up the active site of the um, enzyme are such that they are just complementary to the chemical groups on the substrate, and therefore there can be non-covalent interactions very easily between the substrate and the enzyme. But enzymes are very sensitive to alteration in the chemical group in the substrate, to even slight change in orientation of the chemical groups of the substrate. So we will learn about the different forms of specificity very soon. There you will come across how specific enzymes in nature are. Okay, so the active site is highly specific to certain substrates, meaning it can only bind molecules with a particular shape and chemical structure. Regarding complementary shape, I told you this is like the lock and key mechanism that we have studied, lock and key mechanism of enzyme binding. So the active site has a shape that is complementary to the shape of the substrate it binds. It allows the substrate to fit snugly into the active site. Coming to the catalytic residues. Okay, so within the active site there are certain residues which are mainly involved in binding with the substrate and performing the catalysis reaction so that the substrate can be converted into the product. Okay, so within the active site, see these amino acids, they directly participate in the chemical reaction. Okay, and then comes in what is induced fit model. So I'm sure you know about this. So what happens in induced fit model is that initially the enzyme active site is not exactly complementary to the substrate binding site, but then what happens when the substrate binds to the enzyme, the active site undergoes conformational change and it becomes exactly sufficient to bind to the substrate. So it optimizes the interaction between the enzyme and the substrate. Okay, so if you look at this figure, this is the protein chain, this is your active site, and here this is the active center where all the chemical groups of the amino acids are present, and this is the binding group, this is your catalytic group, and here is the substrate molecule which is binding. Okay, so the characteristics of the active site collectively contribute to the efficiency and specificity of the enzyme reactions by providing a suitable environment for substrate binding and catalysis. Excuse me. So this is the mechanism of enzyme action. So let us look at this. This is an enzyme, and to this enzyme, to the active site of the enzyme, we can see that this particular substrate molecule has been bound. Okay, then what happens? This is converted into product. So the rate of reaction from enzyme plus substrate to this enzyme substrate complex, this rate constant for the reaction is denoted as K1. The rate constant for the backward reaction is denoted as K minus 1. Then what happens? The enzyme and the products are freed and the enzyme goes for another cycle of catalysis. So what happens? This forward reaction, the rate constant is called K2, and we will learn a lot of different formulas where we will see the significance of K2, we will see the significance of enzyme substrate complex. Okay, so let us move on. Now we will talk about the properties of enzyme catalysis. So I told you that initially the reactants, if you look here, the reactants are at a higher energy level compared to the product. So here is the energy level of the product, here is the energy level of the reactants, and this is the energy, this is the amount of energy required for the reaction to happen. But then when enzyme comes into play, it lowers this activation energy up till only this level. Then what happens? This is the lowered activation energy, and therefore it allows the substrate to bind to the product, and it allows the substrate to get converted into the product at a, even if the energy is low, and therefore it speeds up the processes that are essential for life. Okay, moving on. So this is another property of enzyme catalysis. Now enzymes are very sensitive. Okay, as you know that they are proteins, and proteins assume different charges at different pH, and therefore what happens is the protein, the enzyme, it actually remains active only at a specific pH range. Beyond that pH range, what happens? The amino acids in the active site, they assume different charges compared to the uh, specific pH range for optimal activity, and then what happens? The activity is not optimum anymore. Okay, coming to temperature, what happens? At lower temperature, the molecular vibrations are not sufficient, and therefore the rate of the reaction is slower. But at higher temperature, what happens? The protein gets denatured and it unfolds, and therefore once again the temperature decreases. So the temperature or pH at which a substrate shows its maximum activity, this is the one that is called the optimal temperature or the optimum pH respectively. A temperature or pH more than the optimum may alter the molecular structure of the enzymes. Generally, an optimum pH for the enzyme is considered to be ranging between 5 and 6. Okay, so as I told you, because of heat, what happens? The increased energy level of the molecule disrupts the bonds in the enzyme and between the enzyme and the substrate. So that includes hydrogen bond and ionic bonds. Hence denaturation results in loss of the 3D structure of the enzyme molecule. Okay, but then cold renders the molecules slow, decreasing their collision rate, and therefore uh, the reaction rate slows down. Okay, enzyme activity also slows down. Okay, so these are the different factors that affect enzyme activity. They are temperature, they are pH, they are enzyme concentration and substrate concentration, right? So if you look at these two, this tells you, these two graphs tell you that enzymes are more sensitive to pH change compared to temperature change, right? So see, actually why does this happen? Let's say that a particular enzyme is binding to, let's say, ATP. Okay, now ATP is having these phosphate groups, and so therefore the ATP molecule is going to be negatively charged. Let's say that the active site of the enzyme is having lysine residue and arginine residue, which are positively charged, and these two are going to come and bind. But let's say that the pH is very high. If the pH goes beyond 12, okay, then what happens? Then the positive charge is lost from the amino acids, from the enzyme molecule, and therefore what happens? The active site is now what? Negatively charged, and because of that, what happens is, because of that, the enzyme will no longer be able to bind to the ATP molecule, and this tells you that if there is very high or very low H plus ion concentration or OH minus ion concentration in a solution, then it interferes with the enzyme structure. Alright, I'm sure you've understood about temperature and kinetic energy. So if there is too much of kinetic energy, that basically unfolds the protein, it denatures the protein. Then what happens? Enzyme concentration. So initially, as you increase enzyme concentration, if the substrate concentration is also increased at a similar rate, then what happens? More enzyme molecules bind to more number of substrate molecules and the reaction progresses. But then if the substrate molecules are not increased, then what happens? The saturation phase is attained because the number of substrate molecules are limited and all the enzyme molecules are already bound to the substrate molecule. Now even if you add new enzyme molecules, the reaction is not going to proceed, right? Then what happens? There is substrate concentration. It's a similar logic. As the substrate is increased initially, all the enzyme molecules are going to bind to the substrate molecules. They're going to get saturated, and then what happens? Gradually it undergoes a stationary phase. So increase in the substrate concentration initially increases the rate of the reaction until the active site of the enzymes are used. Okay. So finally we will be talking about the properties of enzyme catalysis that also include specificity. So enzymes are very specific and they bind to substrates with distinct chemical groups only in certain orientations. So the different types of enzyme specificity are described below. They are absolute specificity. So the enzyme will catalyze only one reaction. There can be group specificity where the enzyme will only act on molecules that have specific functional group such as amino, phosphate or methyl groups, right? Then there is linkage specificity. So here what happens? The enzyme will only act on a particular chemical bond regardless of the rest of the molecular structure. Then comes in stereochemical specificity. So in case of stereochemical specificity, what happens? The enzyme will act on a particular steric or optical isomer. So this figure gives you a list of the different kinds of specificity that an enzyme can show. These include bond specificity, group specificity, substrate specificity, optical specificity, geometrical specificity and cofactor specificity. Since in today's MCQs we do not have too many questions on specificity, therefore I will not be going into the details of these different types of specificity. But if you want to know more, you can definitely look up. And for this chapter enzymes, please go through the two books that I have added at the end of the PPT. So they are Voigt and Voigt and Leninger's Biochemistry. So these two books are sufficient to teach you, to give you a very good idea about enzymes. But for your examination, for all the entrance exams, all the competitive exams, make sure you practice numericals, because in every single competitive exams they will ask you numericals on enzymes, enzyme kinetics and inhibition and all of these different topics. So make sure you practice those numericals properly. Okay, so let us look at this diagram. Here we can see that enzymes are even specific to the stereo to the optical isomerisms, right? So stereoisomerism. So if you look at this particular figure, this is starch. Starch is made up of glucose molecules and the glucose molecules are bound to one another by alpha 1,4 linkage. Here you see alpha amylose. Now alpha amylase is an enzyme that is going to bind to this alpha 1,4 linkage of glucose molecules, but then it is not going to bind to this beta 1,4 linkage which is seen in cellulose of the same glucose molecules. So this tells you that in the active site the conformations of the amino acids is such that this tiny oxygen actually fits into it, but when the oxygen is in beta position it is not going to fit into the active site, and therefore alpha amylase is not going to act on cellulose and break it down to glucose. All right, we'll move on to some numerical concepts. They are turnover number and specific activity. So I will recommend you to go through all of these in details. So turnover number or molecular activity tells us how many maximum number of molecules of substrate can be converted into a product per catalytic site of a given concentration of enzyme per unit time. So this is defined as Kcat is equal to Vmax by Et. Et is total enzyme. Okay. So the units of turnover number or Kcat are moles of the product per second divided by moles of the enzyme or second inverse, right? So this gets cancelled out and therefore we are left with second inverse. So the number of molecules of the substrate transformed to products by enzymes per unit of time is defined by enzyme activity. This is another term. This tells you that the number of molecules of the substrate converted to products per unit time, that gives you enzyme activity. Then comes in specific activity. Now this takes into consideration the amount of the total enzyme present. So this tells you the activity of an enzyme per milligram of the total protein. Okay. Now if you look at the units, so these are the classical units, these are the international units, and you can see that the unit of enzyme activity is micromole substrate transformed per minute, and that is also written as 1 unit. Okay. For specific activity, it is micromole substrate per minute per mg of enzyme. So that is unit per mg of enzyme. For molecular activity, micromole substrate per minute per micromole enzyme. So that gives you units per micromole of enzyme, and this is how all of these are interrelated. Similarly, when you take this the amount of substrate in Catal. So, number of moles of substrate per second, that is taken as Catal, right? So, you can find out the relation between Catal and micromole of substrate, and then you can try to equate the two units. Okay, for specific activity, it is again Catal per kg enzyme, and for molecular activity, it is Catal per mole of enzyme. Okay, so I'm sure this is clear. Right. Now we will try to solve the numericals on enzyme kinetics. So this is for practice. These kind of numericals are very direct numericals, but they are going to help you to understand what kind of formula needs to be applied and what exactly does the question say. So it's very good for practice, and I would recommend you to practice as many numericals as possible so that you become very, very well versed with these topics. Okay, so 10 micrograms of carbonic anhydrase and the molecular weight is given in the presence of excess substrate exhibits a reaction rate of 6.82 into 10 to the power 3 micromole per minute at substrate concentration is equal to 0.012 molar. The rate is 3.41 into 10 to the power 3 micromole per minute. So first thing that they have asked is what is Vm. Then they have asked you what is Km. Km is your Michaelis Menten constant, right? And what is K2 or Kcat? This is the turnover number. Okay, K2 is the rate constant for the reaction that goes on between ES to E and P, right? So let us look at how this can be solved. So see Vmax you can take out. So the Vmax, half of Vmax is the rate of reaction at which substrate concentration is equal to Km, and therefore from there you can derive this Km, Michaelis-Menten equation. From the Michaelis-Menten equation you can derive Km. Okay. Then we have to divide Vmax by micromole of total enzyme to find the Kcat, okay, or the turnover number, and that is how you can calculate this. Fine. Then we will move on to allosteric enzymes. So what are simple enzymes and what are allosteric enzymes? So simple enzymes are more often unimolecular. Okay, so it is having a single it is a very simple structure, so it is not having too many subunits, and this is it even follows this Michaelis-Menten curve. So in the Michaelis-Menten curve, you can see that when the substrate binds to the active site, then the enzyme becomes active. So the velocity of the enzyme when plotted against substrate concentration, this gives you this hyperbolic curve. But then for allosteric enzymes, if you look at this figure, there is this active site to which the substrate binds. Besides that, there is one tiny regulator site. So this is the regulatory site where what happens, this effector molecule comes and binds. So since this is away from the active site of the substrate, this is called the allosteric site, where if this effector molecule binds, the enzyme undergoes a conformational change, because of which it can show very high activity. Okay, now this effector molecule, this can be either an activator or it can be an inhibitor. So if it is an activator, then we see this graph. So initially the enzyme activity was low, and then the effector molecule got bound, and then the enzyme activity suddenly increased. So this is giving you a sigmoid curve, right? But then if this is an inhibitor, then what happens? The molecule, the activity, the catalytic activity of the enzyme decreases rapidly. Okay, so an allosteric enzyme changes its activity or function in the response to the binding of a regulatory molecule at a site other than the active site. This regulatory site, known as the allosteric site, is distinct from the active site where the substrate binds. Binding of a regulatory molecule to the allosteric site induces a conformational change in the enzyme structure, which can either enhance or inhibit the enzyme's catalytic activity. Fine. So now these are again some terms that you need to go through to understand an allosteric enzyme. So one important characteristic of the allosteric enzyme is its regulatory site, which is also known as the allosteric site. So let us look at it. Okay. So the allosteric enzymes possess an allosteric site where regulatory molecules called the allosteric modulators can bind. These modulators can either be activators or they can be inhibitors, right? So what about conformational change? Yes, so binding of the allosteric modulator induces a conformational change in the enzyme structure, altering its catalytic activity. This change can either increase or decrease the activity of the enzyme. Then comes in a concept called cooperativity. So cooperativity means encouragement. Okay. So what happens? In this case, it means encouragement. So basically what happens in cooperativity is, let's say one particular molecule comes and binds to one subunit of the enzyme. And then what happens? This is a characteristic seen in allosteric enzyme. The other subunits are now prompted to bind to that particular molecule as well. And then what happens? All these molecules together are now bound to the different subunits of the enzyme, and then the activity is improved. Okay, so let's look here. Allosteric enzymes often exhibit cooperativity where the binding of a molecule to one subunit affects the binding or activity of subsequent substrate molecules or enzyme subunits. So if this is a activator, it is going to rapidly increase the enzyme activity because all the subunits are now getting activated. However, if this is an inhibitor, then it is going to be just the opposite. Okay. The fourth characteristic of allosteric enzymes is that they are multimeric in structures. So of course, if you understand, there needs to be an active site, there needs to be an allosteric site, there needs to be so many subunits that make these allosteric enzymes huge, right? So they are made up of multiple subunits, each containing both active and allosteric sites. So binding of a regulatory molecule to one subunit can affect others through cooperativity. Okay. What about feedback regulation? So what happens in case of feedback regulation is you've understood that these enzymes are very catalytically they're very active. Now what happens, the body always has a balance. So balance is it is a property of nature, and therefore what happens in feedback regulation, let's say a lot of the substrate is getting converted into the product, then what happens, this product is now acting as an inhibitor, and it comes and inhibits the enzyme from further activity. So this is called product inhibition, and this is a kind of feedback regulation. So see, allosteric enzymes often participate in feedback regulation where the end product of a metabolic pathway regulates an enzyme earlier in the pathway. Alright, so many enzymes are acting in a metabolic pathway for the formation of the end product, and this end product come and inhibit one of the enzymes, therefore stopping the entire pathway and regulating its own overexpression. Okay, now we will move on to the kinetics of allosteric enzymes. So allosteric enzymes are in R state or high activity states, and they can be in low state or the low activity or T state, right? I'm sure you have learned about this relaxed state and tense state in the hemoglobin molecule while learning about the activity of hemoglobin. Okay, so both enzyme states are present, and then they interconvert in solution. So if you look at this gives you information about the T states. So measured activity reflects both R and T states, leading to a sigmoidal curve. Okay, so this is the activity of the R state, this is the activity of the T state, and because of these two we get the T-shaped curve. When activators are there, more of R state is seen. When inhibitors are there, more of T state is seen, and therefore this is the uh, shift in the sigmoid curve that we see. Okay, now we will talk about inhibition. So what happens in case of inhibition is that there is regulation of the allosteric enzymes. So the allosteric enzymes undergo a regulation in different ways. There can be inhibitor molecules which can bind to the allosteric enzymes. Now, inhibitors can be of various types. They can bind to different sites, and they can also bind to the enzyme at different time points in its catalytic activity. Okay, so if the inhibitor looks exactly like the substrate, or it is having some kind of chemical group with which it binds to the enzyme, and it is very similar to that of the substrate, then it is known as a competitive inhibitor. This competitive inhibitor binds to the active site of the substrate, and then what happens, when the competitive inhibitor comes and binds, the substrate can no longer bind to the enzyme, and therefore it is basically competing with the substrate for the active site of the enzyme. Okay, that is one thing. Now we will look at inhibitors that do not bind to the active site of the enzyme, but then they prevent the catalytic activity of the enzymes. So if you look at this figure, this is a non-competitive inhibitor which is binding to this inhibitor binding site, and it is binding to this particular enzyme substrate complex. So see, inhibitor binds to an allosteric site of an enzyme substrate complex, causing inactivation. So it is binding when the enzyme substrate complex is formed. So for uncompetitive inhibition, inhibitor binds to an allosteric site of an enzyme substrate complex, causing inactivation. So let us look at this in higher details. Okay, all right. So for non-competitive inhibitors, if you see that when this inhibitor, it is already bound to the enzyme, then what happens? Yes, so the inhibitor binds to the enzyme. Okay, so then what happens, the substrate can bind, but catalysis is no more possible. Similarly, this particular inhibitor, it can also bind to either the free enzyme or it can bind to the enzyme substrate complex. Okay, in both the cases this inhibitor binds. So the inhibitor binds to the enzyme or the enzyme substrate complex at an allosteric site, and the catalysis is prevented due to a change in the enzyme's conformation. So because of the change in conformation of the enzyme, if you look at this figure, here the conformation of the enzyme was like this, but here the conformation of the enzyme at the active site, if you see, this is altered, and therefore this is inactive. So it is not sufficiently interacting with the substrate. Similarly, here also, after binding with the inhibitor, the active site conformation here, if you see, this is altered, and therefore even if the substrate tries to bind, there will be no catalysis. Fine. Now, for uncompetitive inhibitor, the inhibitor can only and only bind to the enzyme substrate complex. They will not bind to the free enzyme. Okay, so the inhibitor binds to the allosteric site of the enzyme but not to the free enzyme. The inhibitor is not structurally similar to the substrate and does not compete with the substrate for the active site. Okay, now these are the two differences between the uncompetitive and non-competitive inhibitor. And then you can also study about mixed inhibition. That would be a very interesting topic to get to know. Okay, now we will look at some plots in enzyme kinetics. Now this and the numericals are the most important properties, are the most important parts of this particular topic, and I recommend you to go through the books that I have recommended. There are a lot of websites which you can refer to. I have collected all of these figures, all of the theory portion from the websites and from the books, so please refer to all those. That will give you a very good idea of the entire subject. So since Vmax is achieved at infinite substrate concentration, it is impossible to estimate Vmax and hence Km from a hyperbolic plot. Okay, so if you look at this Michaelis-Menten curve, you see that this is going into saturation, and therefore it is next to impossible to find out the real value of Vmax ever, right? Because this is a hyperbolic curve. It is going into an infinite. Even if we go to infinite substrate concentration, Vmax, determining the actual concentration of Vmax would be difficult. Therefore, because of this difficulty, the Michaelis-Menten equation was transformed into an equation for straight line by Lineweaver and Burke. Okay, so 1 by reaction rate, or 1 by V was taken and 1 by substrate concentration was taken, and then this is a double reciprocal plot that has been performed. So reciprocal of the y-axis coordinate, reciprocal of the x-axis coordinates, and therefore this is double reciprocal, and therefore we get this straight line, and the point on the y-axis where it intersects is 1 by Vmax. The point on the x-axis where it intersects is minus 1 by Km, and the slope gives you Km by Vmax. Okay, so the Lineweaver-Burke plot or the double reciprocal plot is a graphical representation of Lineweaver-Burke equation of enzyme kinetics. Okay, so this V is the initial velocity or the rate of the enzyme reaction. Vmax is the maximum velocity or the rate of the enzyme reaction under the saturating substrate concentration. Substrate concentration is represented as this. Km is the Michaelis constant, which represents the substrate concentration at which the reaction rate is half of Vmax. So this is your Km. This is half of Vmax, and the substrate concentration at which half of Vmax is attained is called the Km. Right? Now there are certain assumptions in the Michaelis-Menten equation. So there are basically 5 different assumptions. Okay, so let us have a look at them. This is E plus S, that is enzyme plus substrate, and this is the forward reaction, this is the backward reaction, that gives you ES. Then again there is a forward and backward reaction, and the rate constants are K2 and K minus 2, that give you E plus P, that is enzyme plus product. So assumption 1 is that there is no product present at the start of the kinetic analysis. So here, when ES is present, there is initially no product. And therefore, we can omit this particular reaction, that is the backward reaction. Right, so we get this. See here, therefore, as long as we monitor initial reaction rates, we can ignore the reverse reaction of E plus P going to ES. Okay. Then there is assumption 2. So during the reaction, an equilibrium condition is established for the binding and dissociation of the enzyme and substrate, that is Briggs-Haldane assumption. Thus, the rate of formation of the ES complex is equal to the rate of dissociation plus breakdown. Okay, so this tells you about the steady state equilibrium, and this tells you that the rate of formation of the ES complex is equal to the rate of dissociation plus breakdown. Alright. Okay. So assumption 3 is that the amount of enzyme present is much less compared to the amount of substrate. So the enzyme is a catalyst, and therefore it is recycled again and again. Therefore, only very small amount is required. The amount of substrate bound to the enzyme at any given moment is small compared to that of the substrate. So, it follows that the enzyme substrate complex again, this is also very small compared to the substrate, and therefore S is constant, substrate concentration is constant during the course of catalysis. Okay. For the fourth assumption, only the initial velocity of the reaction is measured. So, the product concentration initially is 0, and substrate concentration is much more than the initial substrate concentration. For the fifth assumption, the enzyme is either present as a free enzyme or as ES complex. So, amongst all the enzyme that is there, a part of the enzyme is involved in the formation of the ES complex, and the rest of the enzyme is present as E free enzyme. Okay. So, this again tells you that the maximum possible velocity or Vmax occurs when all the enzyme molecules are bound with the substrate, that is ES is equal to E total. So, all these points that are written here, these are very important for the MCQs that are coming up. Okay, and therefore I tell you to please go through the slides, to go through all the websites and everything given as references, because these are also there in your questions. All of that is written here is very important for your questions. Okay. So Vmax is equal to K2 into E total. This is an important formula. So this K2 is nothing but K cat, and you know that K cat is equal to Vmax by E total. This is the formula that we learned for turnover number just a few slides back. So substituting this for the Michaelis-Menten equation, we get this, and this is the mathematical representation that is used to model the experimental kinetic data. Now, Km is given as K minus 1 plus K2 divided by K1. So, that is rate of breakdown of the ES divided by the rate of the formation of ES. Okay. So, Km is similar but not exactly equal to a dissociation constant or Kd of the ES complex. So, all of these, whatever points we are discussing right now, these are very important for the MCQs. Okay. So, now they are telling you that Km is basically similar to, but it is not exactly the dissociation constant for the ES complex. Okay. So, if K1 is much greater than K2, so K1 is the forward reaction from E plus S to ES, then Km is much greater than Kd. Okay. Due to this similarity to the expression for Kd, a low value of Km is often interpreted as high affinity of enzyme for the substrate, and a large value of Km is often interpreted as a weak affinity of the enzyme for the substrate. Okay. So this is very important. This tells you that, this gives you the reason why exactly do we associate Km with the affinity between the enzyme and the substrate. So the answer is given here. That is because at a low um, at a low value of Km, it is similar to Kd, right? And it is acting as the Kd. So now if you see here, Km has units of molar concentration just like the unit of substrate concentration. Fine. So there is a mathematical treatment that allows for the determination of Km from the experimental V versus concentration of S data. Consider the situation where the concentration of S being evaluated results in the value of V that is exactly half of the maximum reaction velocity. So if you substitute this V for half of V max and if you solve it, then you come to this conclusion Km is equal to S. So that tells you that Km equals to the substrate concentration that results in exactly 1/2 maximum velocity of the reaction. So now you understand why Km is equal to the substrate concentration at half of maximum velocity. Okay. So now we will look at the Michaelis-Menten plots for different inhibitors. Okay. So how this particular Michaelis-Menten plot changes when different kinds of inhibitors are added to the enzyme substrate reaction. So if you look at this, this is the original plot for the free enzyme without any inhibition. Let's say that Km is 10 and Vmax is 20. Okay, so Km is just 10 and V half. So Vmax here, this is 20. Fine for free enzyme. Then you add a competitive inhibitor. So what happens when competitive inhibitor is added? The inhibitor is going to come and bind to the active site of the substrate. Therefore, the affinity of the enzyme towards the substrate, it reduces, and if the affinity reduces, then Km is going to increase, and therefore you see Km becomes 20, right? So now this is the Km. So Km has increased, but your Vm, it remains constant. Okay. Then it is non-competitive inhibitor. So what happens? Non-competitive inhibitor comes and binds to the allosteric site. Then what happens? It allows the substrate to bind, but then it prevents the reaction to proceed. Substrate can peacefully bind, but then the reaction would not proceed, and therefore you see that the reaction is going at a very low rate, which is Vmax is equal to 10. So this is reduced, but then Km is unaffected. So inhibitor reduces enzyme activity but does not bind to the active site. Okay. Now we will move on to this Lineweaver-Burke plot. So what happens in case of Lineweaver-Burke plot? 1 by V is equal to Km plus substrate concentration divided by Vmax into substrate concentration. So that gives you Km by Vmax into 1 by substrate concentration plus 1 by Vmax, right? So this is the Lineweaver-Burke plot and this is the equation. So if you look at the graphs, this is the one for competitive inhibition. Here what happens? This is your 1 by Vm. So 1 by Vm, you can see it is constant. Whether the inhibitor is there, whether there is no inhibitor, it is constant. But Km is actually increasing, right? It is becoming less negative and therefore it is increasing. So Km increases and Vmax remains unaffected. The slope changes, y-intercept is same. So slope is changing, but then the y-intercept is the same, correct? Then we will see uncompetitive inhibitor. So what happens in uncompetitive inhibitor? Km decreases, Vmax also decreases, right? So this is 1 by Vmax and therefore, yeah, it's going to decrease. So this tells you that Km gets reduced, Vmax gets reduced and slope remains the same, but the y-intercept changes. For non-competitive inhibitor, Km value is exactly the same, but then Vm value, you can see that this has decreased, right? So Km is unaffected, Vmax is reduced. Slope changes and y-intercept also changes. Also for mixed inhibition, this is also the case for mixed inhibition. Okay. Now we will start with the week 8 assignments. So all the questions have been covered here for week 8 assignment. Let us go through them quickly. So in the first question, it is a question on enzyme kinetics. Right? So let us try solving it. So for a given enzymatic reaction, S plus E gives you ES, that gives you E plus P1 should monitor the absorbance of dash versus time. So complete the sentence. The options are product, substrate. Substrate is going to reduce, so it is not going to be the case. Then tyrosine or tryptophan residue present in the enzyme. So this is not going to be the case because enzyme is going to be exactly the same, right, all through the reaction. Tyrosine or tryptophan residue present in the active site of the enzyme. Once again, you cannot do that. Fine. So the right answer would be the product. Then we will check this. For the reaction, the same reaction is given. They are asking you which of the quantities can be measured experimentally. So let us see substrate, product and total enzyme. Total enzyme you can measure. Substrate, amount of substrate also you can, you are adding. So you can measure product, whatever is formed, you can measure that. Then the bound substrate. So this is impossible to measure, right? So bound substrate you cannot measure. Bound product also you cannot measure. Same with bound enzyme, and so therefore you cannot measure this. Only substrate and product. Okay, you can also measure total enzyme, right? So this would not be the case. Only free product. No. So the right answer should be A. Let us look at the right answers. Yes. So the first option is the right answer in both the cases. Fine. So for the next question, we will see when the velocity of enzyme activity is plotted against substrate concentration, which of the following shape is obtained? Right. So this is your velocity of enzyme activity plotted against substrate concentration. So let's see, this is substrate concentration and this is the velocity of the reaction. I am sure you remember this was your Michaelis-Menten equation, and therefore this is it a parabola? No, it does not look like a parabola. Is it a hyperbola? Yes. Straight line with positive slope? No. Straight line with negative slope? No. So therefore the right answer should be B. This is a hyperbolic graph. Yes. So if you look at this, this is the rate of the reaction on the y-axis, the substrate concentration is on the x-axis, and this is initially the rate of the reaction, and then it goes to saturated phase when all the active sites are occupied. Here more active sites getting filled, and therefore the reaction is increasing. Rate of reaction is increasing. Okay. So for the next question, this is a very direct question. It's a match the following. So I'm sure you remember that enzymes are divided into 6 different classes. So they are oxidoreductases, lyases, ligases, hydrolases, and there are also 2 other classes. So you can read about this. I'm sure you all have learned this during your school time, high school, and also possibly in college. So see which of these particular enzyme classes are doing what kind of function. So if you look at this one, hydrolases, what is it doing? It is going to add water across a bond, hydrolyzing it. So this should be 2. So the fourth option should be 2. Okay. So what about, so this is basically your 2. Now what about ligase? Ligase catalyzes reactions in which 2 chemical groups are joined using ATP. So it should be the first option. So let us look here. So 4 is 1 in all the 3 cases. So it would not be B because 4 is 2 here. So it can be either this, it can be this, and it can be this one. Another one is hydrolases. It should be 4. So 2 should be 4. Yes, 2 is 4 in only this one. So this should be the right answer. Let us see other options. oxidoreductases. Oxidoreductases, what does this do? They add or remove hydrogen group. Yes, so this should be this one. And then we are left with lyase, which adds water, ammonia or carbon dioxide across double bonds or remove these elements to produce double bonds. Okay, so let us look at the answer. Yes, it is B. Okay, right. So we will move on to the next two questions. So in which form the enzyme exists when the maximum velocity is observed? So we learned previously while talking about the different assumptions and different formula that maximum velocity is attained when the enzyme is in the ES form. Okay, so you can refer to the PPT and confirm that this is the case. Then in the enzyme kinetics, turnover number is defined as maximum number of substrates that can be converted to the product per mole of catalytic site per second. So you know that turnover number is equal to Vmax by Et. So concentration of the enzyme required to reach maximum velocity, number of enzyme substrate complexes in the reaction, concentration of the enzyme required to reach half the velocity. Once again, not the right answer. Right? It is not the concentration of the enzyme required to reach maximum velocity. So the right answer should be option A. Okay, and for the above question, it should be option B. Now we will move on to another question. Which of the following is true regarding the Michaelis-Menten kinetics? So Km, the Michaelis-Menten constant, is defined as the concentration of the substrate at which enzyme is working at maximum velocity. No, that is wrong because enzyme is working at half of the maximum velocity. So this is half of the maximum velocity. Correct? It describes single substrate enzymes. Okay, that is one thing. And then came the Michaelis-Menten constant is defined as the dissociation constant of the enzyme substrate complex. It is not defined as the dissociation constant, but it can be compared to the dissociation constant only when the value is very low. So only when the concentration of the ES complex is very low, it can be compared to the dissociation constant. So see, let us look here. Michaelis-Menten kinetics assumed covalent binding between enzyme and substrate. So there is never covalent binding between enzyme and substrate. It is always comparatively weaker interactions. So therefore the right answer would be B. So Km is defined as the concentration of the substrate at which the enzyme is working at half of its maximum velocity. It is also a measure of affinity that the enzyme has for the substrate. Michaelis-Menten kinetics assumes non-covalent binding between the enzyme and the substrate. Okay. So for the next two questions on enzymes, we will be looking at these two questions. Which of the following is not a characteristic of the enzyme? So enzymes accelerate reactions by increasing free energy of activation. Increasing the free energy of activation, that is not the case. They decrease the free energy of activation, so this is wrong. Enzyme bind to the transition state of the reaction better than the substrate, that is also not correct. Enzyme exhibits saturation kinetics, which is true because you know that the graph goes like this, right? So if that is the Michaelis-Menten curve, then it goes like that. Enzyme works best at optimum pH and temperature. That is also true. So this is correct and this is definitely not correct, and so your this A should be the right answer. Enzyme often bind to the transition state of the reaction better than the substrate. This is also possible. So then the right answer would be A. So small organic molecule bound to the apoenzyme by a covalent bond is called the prosthetic group. It is not a complex enzyme, not a hollow enzyme, and coenzyme does not bind using covalent bond. Okay. So if you look at this diagram, okay, we will take a small break of we will take a very small break of 15 minutes and then we will be back again with the questions. So I have a few more questions and then we will be done. We will also keep learning the different topics as we go about with the questions. Okay, we will learn the topics as well. Okay, so let us return and let us resume with the questions. Okay, so this is a question on enzyme inhibition, and you can see that in this case, yes, so in this case the substrate is binding to the enzyme and your inhibitor is also binding to the enzyme, and it is binding to the active site of the enzyme. It is not binding to some allosteric site. So this confirms that this should be which one? Uncompetitive? No. Should it be non-competitive inhibition? No. It is also not allosteric inhibition. Rather, it is competitive inhibition. Right? So yes, that is the right answer. This is competitive inhibition. Fine. So in the next question, this is a question given on Lineweaver-Burke plot for any enzyme, and it is showing you two different plots, two different lines. One is for without inhibitor, and then there is a shift when the inhibitor has been added, right? So what do we see? This is your 1 by Vmax initially, this is the 1 by Vmax later. So that tells you that if 1 by Vmax has increased, that means Vmax has decreased. So that tells you that this Vm has decreased. Besides that, this is your 1 by minus Km, and this is 1 by minus Km after adding the inhibitor. So since this is increasing in the negative axis, we can say that Km is also decreasing, and therefore this kind of graph, we do not see it in we do not see it in case of competitive inhibition, because in competitive inhibition Vmax remains unchanged, right? For non-competitive inhibition, also we do not see this kind of a graph, right? Because there your Km remains unchanged, but for uncompetitive inhibition, both of these are reduced, and therefore this is uncompetitive. For mixed inhibition also, this is going to be the case in case of mixed inhibition, and therefore the right answer here would be uncompetitive inhibition. Fine. So now we will talk about chemistry in fruits. So when a sliced apple is kept in open air, it becomes brownish. And what do you think causes that? So I'm sure this phenomena we have experienced so many times, and I'm sure you might have been curious what exactly is going on inside the apple that is making it brown. Okay. So oxidation of phenolic compounds in an apple by an enzyme O diphenol oxidase. If that is the case, okay, is that the case? Then again, in the second option, we have reduction of the phenolic compounds in apple by enzyme O diphenol oxidase. Then there is inhibition of the enzyme O diphenol oxidase. And finally, we have inhibition of phenolic compounds in the apple by enzyme O diphenol oxidase. Okay. So if you look at the answer, when an apple is cut into pieces, the plant tissue is exposed to oxygen. Therefore, it should be oxidation. And so this triggers an enzyme known as polyphenol oxidase, or PPO, that you can see here, PPO, to wait for it to oxidize polyphenol in the apple's flesh. This results into a new chemical called orthoquinones, which then react with amino acids to produce brown colored melanin. So this brown compound is nothing but melanin. It is happening when aerobic oxidation of phenol by this polyphenol oxidase to give your quinone, and then that quinone is reacting to give melanin, right? So quinone is reacting with amino acids to give melanin. So that is because of aerobic oxidation, and therefore this should be the right answer. Number A should be the right answer. Fine. For the next two questions, they are asking you to fill in the blanks. Okay. So in an enzyme catalyzed reaction, dash provides information on substrate binding and dash provides information on reaction rate. So for substrate binding in an enzyme catalyzed reaction, it is Km, and Vmax provides information about the reaction rate. So C should be the right answer. Then for the following equation, Line-Weaver-Burke plot, you tell me which one is Line-Weaver-Burke plot. It is double reciprocal plot, right? So this is your Michaelis-Menten equation. So this is not the case. Then which one do you think is the case? Well, it is D. Okay, so D and C, these are the two right answers, and this is the equation for Line-Weaver-Burke plot, which is a double reciprocal plot. You can go back to the other slides in the PPT to confirm. Okay. Finally, we move on to the last question of this session, which is on RNase A. So which of the following statements is or are correct regarding RNase A? So this is a very direct question, and for this you will have to study about the catalytic mechanism of RNase A. So RNase A catalyzes the cleavage of the phosphodiester bonds by formation of trivalent phosphate transition state. RNase A catalysis histidine 12 and histidine 119 play an important role, and they have the ability to both accept and donate electron. The specific substrate for RNase A is cyclic CMP. Phenylthiourea is the standard inhibitor for RNase A. Okay, so which one do you think is the right answer? Okay, so if you look here, the right answer is D. So 2 and 3 are correct. So these two are correct. 4 and 1 are not correct. So let us see why that is the case. RNase A catalyzes the cleavage of the phosphodiester bonds by the formation of pentavalent phosphate transition state. So they have mentioned here trivalent. That is not the case. It should be pentavalent, right? Phosphate transition state. The histidine 12 and histidine 119, these are two residues that have the ability to both accept and donate electron. That is true. So that means this is correct. Then the specific substrate for RNase A is cyclic CMP. This is also correct. Right. So let us have a look. Let us have a closer look at RNase A now. So this is the scene at the active site of RNase A. This is an RNA molecule, and initially this histidine 12 is acted as a general base, and histidine 119 is acting as an acid to promote nucleophilic attack and bond cleavage. So you can see that this hydrolysis is happening, and 2,3 cyclic nucleotide is formed. Then what happens? Histidine 2 is now acting as an acid, and histidine 119 is now acting as a general base to promote the hydrolysis, right? So what happens? This attack is now on this 2,3 cyclic monophosphate, and what happens? The interaction with the histidines are lost, and this is released, right? This is how it cleaves RNA. So these are the different books. These are the two books that you can refer to. These are the different websites which you can have a look at. And thank you for watching the video till the end. All the best for your future endeavors.